before, but I put the windows back on because there was one program for the Corona Detective, the other machine that I needed was the Windows system. So what I really wanted to put both of them together, but that's the thing I haven't figured out. So I just thought for this trip, yeah, no, one after, like one inside the other. I think it's possible.
Or you can switch.
But I haven't, yeah, then you can switch. And the real idea of Hackuarium is that we're going to democratize research. And so today I was planning to do the experiment for Corona Detective, and 4 of you gave me samples. But one of the buffer solutions was we didn't have enough of it, and then I lost the tube. So and the incubation is a bit of a challenge for the temperature. So I'm going to go ahead and do the experiment at lab. But basically, seeing the invisible is one of the big aims of understanding our world, because the closer and closer you look at things living, the more beautiful they become. And the closer and closer you look at some of these man-made things, like, say, my soldering, the more ugly it becomes. And so the idea that you can look at microbes, like all these invisible microorganisms that are part of our body on the surface and inside of us that we couldn't be happy and living well without, also you should get an opportunity to explore those. The idea is that this seeing the invisible isn't just this one little presentation, but actually sort of 3-part thing of looking at also the microbiome and making these little paper microscopes, the fold scopes. And this picture shows the ideal output of control reactions for the Corona Detective. I'm afraid on the screen the color doesn't show as well, but it uses a quenched fluorescence method of molecular amplification with enzymes that you can provide as freeze-dried compounds in tubes, so that you can ship it anywhere
that
without
was a lot
needing
of
the
words
cold
in the same
chair.
sentence. What?
So I think it'll be very helpful for really surveillance screening of people without symptoms to find the people who might be better off keeping away from other people, so we can end this pandemic, especially as the vaccinations aren't going as quickly or as well as we'd like. And then the lamp in dark COVID times. So it's loop-mediated isothermal amplification. And so together with a bunch of people from just one giant lab, and especially Guy Edelberg and Fran Caro, who's Guy worked with GMO Detective, which was the real inspiration for this, where he made the detector for GMO Detective, and that's what it says on the front of it.
Maybe you can say what's Joggle.
Yeah, so Joggle is just one giant lab. It's Thomas Landren, who started La Paillasse in Paris, another open community lab. He sort of almost 3 years ago made this online community, and it was especially looking at things like vaccine hesitancy.
For pandemic time, like in 2020 in March, there was this big open call. People gathered and they formed a group. They had Google Sheets, and it was about how many people participated?
There is at some of our calls we had about 200 people on these Zooms,
Yes, so
but lately it's more like 20 or 30 if we're lucky. And so hopefully actually this open science efforts are going to continue more. And the great thing was that they had this new unique form of open peer review. And now we hope it will also be going on beyond Corona. And again, the funding was really essential because at Hackuarium, we're really just funded by members' fees. We don't have, we're all volunteers too. And it's really project-based, and we try to make these transdisciplinary teams with people with all sorts of experiences to get special synergies. And so, we had so many Zooms. I'm totally zoomed out. But this method was really inspired by the GMO Detective. And so I thought it was easy to show the overview. So GMOs are transgenic organisms. Sometimes things like corn or soybean, like 90% of the world's supply of soybean is GMO. And often it's put into milks and cereals and stuff without labeling. And so Guy said, we're going to figure out how to show people to see DNA, seeing the invisible, with this fluorescence detection method. And he did a lot of workshops. He got his PhD from the Centre d'Interdisciplinary Research in Paris just last spring. And so, for instance, corn or soy, I said it, is often GMO, and a very simple DNA extraction, just boiling water, a bit like I did with some of your salivas, lets you do this reaction where all the reagents are dried in the bottom of the tube, and you can put in different controls, so you know you have a negative control and a positive control to know your test was reliable. And after 63 degrees for about an hour, you get the fluorescence or the darkness. And again, this freeze-dried format we think is amazing because you can send it anywhere in the world without cold chain dependence. And we have people in Ghana right now checking over 200 tubes with clinical samples, RNAs. And there they have a real problem. And maybe they're going to get the official regulators to okay this test. And we hope it'll help end this pandemic. So our cells are the building blocks of our organisms, and there's a nucleic acid inside a nucleus in our eukaryotic cells, and each nucleus has lots of chromosomes that are compacted, the code of life. And so here you can see from the cell to the nucleus to the chromosome, and DNA, of course, is like the hard drive, all of the information to make our organism. And through evolution, we still have lots of genes that are related to the, to say, C. elegans, to a nematode worm, and to even bacteria, because our mitochondria were sort of an endosymbiosis from bacteria to help create our eukaryotic organisms. But in terms of corona detective, what's really essential is these very small pieces of DNA. If you have 30 nucleotides in a stretch, it's thought to be basically unique. You'll only find it in one place unless it's just AAATTT. But basically, the nucleotides are made of 4 bases: cytosine, guanine, adenine, and thymine. And they fit together through hydrogen bonds. This base pairing mechanism allows also the replication of DNA. So every time a cell divides, all the DNA is duplicated, and there's a semi-conservative method where each strand, when it opens up, becomes the template for the new strand. They call it semi-conservative replication. And so the way they pair together determines the new baby strand of DNA. And so the central dogma of molecular biology is that DNA goes to RNA, which then encodes proteins. And there's a molecule that incorporates it into the double-stranded DNA. And you always see this late amplification, almost always, unless you have super good primers. So basically, we've gotten it down to 20 copies very reliably, but a colleague in Pennsylvania did a big comparison between our quasar detection and other things called molecular beacons and DARP, which are also similar quench fluorescence type things. And he got down to 2 copies per reaction.